Okay, now is the turn of Cecilia Andreazzi. She's a researcher at the Mammal Biology and Parasitology Research Group at Oswaldo Cruz Foundation in Brazil, and also a member of the Innovation for Resilience, Inclusion and Sustainability Research Group based at the Federal University of Paraíba in Brazil. Her research focuses on understanding the effects of biodiversity loss and land use change on ecosystem functioning and human health at multiple spatial and temporal scales. Her talk is entitled Community Composition and Interactions Determine the Role of Multiple Host Species on the Spread of Zoonotic Parasites, the Ecomultiplex Network Model. Thank you, Cecilia.
Can you, oh no. Can you see my presentation? No.
No, yet no.
Can you see my presentation?
Yes, we can.
Now.
Okay.
Yes, now.
Good evening. So thank you very much. For me, it's a great honor to be here participating in this symposium today. And today I'm going to talk about how the use of multiplex networks help us to study and model the transmission cycles of zoonotic parasites, and also how they may help us in managing healthy landscapes. So zoonotic parasites, they have complex transmission cycles because they circulate among several host species and through several transmission routes. Here I'm just giving three examples of these. For example, the Yersinia pestis, it causes plague in humans, and it can be transmitted through vectors, by air, or by meat consumption. Also, the example of Coxiella burnetii, it causes the key fever, and it can be transmitted by air, ingestion of contaminated milk, or through predation. Cryptosporidium also can be transmitted by the ingestion of contaminated food or by contact with contaminated oil or water. And all these complexities of transmission cycles is what explain some of the apparent contradictory results that have been found in the research that relates biodiversity with infectious disease risk. And the main reason for that is because this relationship, it tends to be extremely nonlinear. So if we want to have a clear picture about how these many anthropogenic processes, such as the climate change, the intensification of agricultural practices, and deforestation, they affect infectious disease risk, we need to have in mind that the temporal and the spatial scales affect this shape of the disease and biodiversity relationship. We also need to understand how the transmission mechanisms and the ecological parameters, here marked with these stars, as for example, the encounter rate, the transmission rates, the host and vector abundances, how these mediate the relationship between biodiversity and disease. We also need to investigate the different dimensions of biodiversity, such as the functional composition of communities and the distribution of species abundances, and not only the species richness. So my interest in the ecology of wildlife diseases started about 15 years ago, when I was still a bachelor student working in this big project that aimed to investigate the effects of parasites on host population dynamics. You can see that at the time we were still using the masks. And during that time, I entered in contact with people working with Trypanosoma cruzi. In this sense, the Trypanosoma cruzi is an excellent system to develop such models and investigate the role of biodiversity on the transmission cycles. Trypanosoma cruzi causes the Chagas disease in humans, and there are two main mechanisms in which the parasites circulate among its wild hosts. One is the vector transmission, marked here with the red arrows, and it involves the kissing bugs. And the second one, that is marked in yellow, is called the oral infection, and is mediated through predation of infected prey, which can happen when, for example, a mammal eats an infected kissing bug or eats another infected mammal. And this is the geographical distribution of Chagas disease in the world. And you can see that Brazil is considered an endemic area and also has a high prevalence of this disease. In Brazil, the vectorial transmission mechanism has been the main route of human infections since the discovery of the Chagas disease. But recently, we have been observing a transition in the epidemiology of Chagas, which is now mainly transmitted by oral infection. You can see here this trend in the map, where there are 3 different time periods are shown. So the P1, the P2, and the P3 with the years shown there. And it's very clear the great increase in the oral transmission that are the red bars, and particularly this is happening in the Amazon region. And I brought the Chagas disease example to this symposium because although it is a disease that was discovered more than 100 years ago, we are now facing this new epidemiological profile, which is highly linked to the intensification of the acai production in the Amazon. In the top map, you can see the number of acute Chagas disease cases in the last 20 years and how it is largely concentrated in the Amazon. So this work, it started some years ago when I met this group of people at the Complex Systems Summer School of the Santa Fe Institute. So at that time, the use of this type of multi-layer networks, they were starting to be used to answer many questions in different complex systems, like the cognitive networks, infrastructure, transportation, and mobility. And our aim was to develop tools to understand the complexity of the trypanosoma transmission on the wild hosts. So a multiplex network is an efficient way of representing systems in which species engage in multiple types of interactions. So in the case of the Trypanosoma cruzi transmission network, we have a basal dispersal layer, that is the geographic position of each species in the environment. The trophic layer includes the predator-prey interactions. It is the food web layer. And the vectorial layer represents the interactions related to the vectorial transmission. So to parameterize the species distribution on the geographic layer, we use data on species body masses and metabolic theory models to estimate the population densities and the home ranges of each species. We use previous data on species diets and serological surveys to determine the structure of the food web and the vectorial layers. I'm showing you the 2 different environments that we modeled. So the canastra and the Pantanal. And you can see that they have different multiplex structures. And these structures, they differ in both layers, in the food web and in the vectorial layer. So you can see the canastra has a much more connected food web layer than Pantanal, and different interactions in the vectorial layer. And just to give you an idea about how we model the transmission dynamics on the multiplex networks is the following. In the left box, you can see the trophic and the vectorial interactions that we can find in each layer of this multiplex. So the trophic is in the top and the vectorial in the bottom. We start the simulation infecting one node, which now became this red square. The probability of vectorial transmission, PV, is a free parameter, and it will determine on which layer the infection happens. If it's on the trophic layer, then the links are directional, and in this case, it's only the blue circle that can be infected. But if it's on the vectorial layer, then the red square can infect any of the other 3 circles. And this is how we begin the parasite transmission simulations. In this plot, you can see that different species are shown in different colors. So we started infecting a number of nodes, which are now colored in red, and each different color is a different species. And as I showed before, we considered a susceptible infected model. So the transmission goes infecting all the next nodes. And it goes until we get all the nodes infected in this environment. And the time to infect all the nodes in the system is exactly the metric that we use for the parasite dynamics on the multiplex. So our first question was to understand what is the importance of each transmission mechanism on the dynamic of the parasite spread. And to answer that question, we compare scenarios with different relative abundances of vectors and also different probabilities of vectorial transmission. So in this plot, you can see in the y-axis is our metric of the time to infect all the nodes in the system. It's called the infection time. And in the x-axis is the probability of vectorial transmission, the PV. And the 4 different colors are the scenarios that we increase the relative abundances of vectors in the environment. We found that when we have a higher abundance of vectors in the environment, then there is a linear relationship between the probability of vectorial transmission and the speed of parasite spreading. And this is more or less intuitive, since we expect a faster transmission when we have a lot of vectors in the environment, and the vectorial transmission is the main route of infection. However, when vector abundances are not so high, then we found that a combination of both the vectorial and the trophic transmission are the most efficient way to spread the parasite in the system. And this is very interesting from an evolutionary point of view, because it explains us why keeping such a complex transmission cycle is efficient for parasite propagation. To investigate how community composition and species traits affect the speed of parasite spreading, we simulated different scenarios in which we humanized groups of species and compared the time to infect all the nodes with the baseline scenario, that is the complete network. So these immunization simulations are like theoretical experiments that are equivalent to removing some species out of the multiplex and then comparing with a complete network. This is some of the results. Each point in this graph represents the increase in infection time when we compare to the baseline scenario, that is the 0, is the complete network. And the x-axis are the scenarios with different probabilities of vectorial transmission, that is the PV. The first thing I want you to notice here, particularly in this lower part of the plot, is that the two communities, the Canastra and the Pantanal, they have different dynamics in these 4 scenarios that I marked with the square. The first scenario is removing all the crucidae species, which are the rodents. And the second scenario is removing all the deer-food species, which are marsupials. The third one is removing all the large mammals, and the fourth is removing the insectivore species. They eat bugs. And for example, you can see that in the Canastra, the orange dots appear below the 0 line, and in Pantanal, they appear above. This shows us that the two communities, they experience different dynamics when we remove the species according to the taxonomic and the ecological traits, such as the body size and diet. This means that it's the local distribution of the traits and the network structure that determine the species' epidemiological roles in the system. Therefore, we cannot make general assumptions about the relationship between the species traits and the epidemiological roles if we don't know the structure of the local communities. But the general trend that we can see is that in both communities, it is the parasite spreading is much faster when we remove the species with the larger body sizes. And these indicators that defaunation and the loss of large predators, they may amplify parasite transmission in these systems. The second general result is that parasite spreading is much slower when we remove the species that had interactions on the vectorial layer, that are the squares in this plot, which we call the ecomultiplex features. This reduction was very similar to the scenarios when we removed the species that showed the highest positive parasitemias in the local studies, the triangles, which we call the epidemiological features. So these mean that the topological role of species on the multiplex are also good predictors of their epidemiological role. And this is super important because it means that we can have insights on local transmission dynamics, even if we don't have data on parasite prevalences. We are currently working with a socio-ecological multiplex in which we included human populations, domestic animals, and villages in the model. The approach we used was to divide our environment in two windows. And there's a different combination of species on each window, which you can see here because they have different colors. But there are species in common that linked both environments. And here are some preliminary results. What is interesting to see is that there's not much difference in the parasite spreading time when we compare if the infection started in the nature or started in the village. And the different colors are the different vector frequencies in the environment. But as you can see here, marked with the red squares, is that humans, they get infected much faster if the spreading starts in the village. Another interesting result is that predators are the group that get infected faster, even if the infection starts in the village. And this corroborates the notion that predators have an important function as bioindicators, and they could be monitored in public health surveillance programs, as, for example, sentinel species that give us early warnings about human infection risk. What I'm doing now, in collaboration with the Janssen Lab, is studying the networks of interactions among wild mammals and the many species and genotypes of Trypanosoma that circulate in different environments. In this map, you can see the great diversity of different Trypanosoma genotypes and how they're distributed across the Brazilian biomes. You can also see here in this smaller map which are the species that carry the highest diversity of Trypanosoma genotypes in each environment. We want to understand how the Trypanosoma and mammal host networks are spatially distributed, and if the topological role of host species vary across this large geographical scale. Here, I'm just showing one example of these networks, which was a study developed at the São Paulo Zoo. We also want to understand if there are host functional traits or functional groups that are more correlated with certain Trypanosoma species or genotypes. We also want to understand how the functional diversity of mammals affect the infection rates across the communities. And now, just to finalize, and thinking about sustainable food production and economical networks, I realized that what we're still missing here in this multiplex are the resources. So the acai palms in this case, they are very important nodes in this system because they are a nesting site for the vectors, they are food resource for the mammals, and they are also important commodity that has a very complex production chain. And this production chain feedback on the entire structure of the landscape. It determines the distribution of the land use. So this gives us a perspective of including trade as an economical layer in this multiplex. Another possibility is also using adaptive networks and let the structure of the geographical layer of the multiplex to change as a consequence of changes in the economical layer. So if we want to build healthy landscapes, we need to think about health as a complex system that is interdependent on biodiversity and ecosystem functioning. For example, in Brazil, it is very clear the relationship between soil production and the incidence of hantavirus infection in humans. And this is very clear, so how the use of landscapes affect human health. And my question here is, can we think about one health in a society that has a production system based on monoculture, cattle, or in oil? In this sense, a great challenge is also to take into account that both biodiversity and health, they are affected by external systems with several positive and negative feedbacks among them. So we urgently need to move to complex system approach to understand health in all its dimensions and also related to all the anthropogenic drivers that are changing the world. With that, I would like to thank you very much and thank all my collaborators and funding agencies. So this work started during the Santa Fe Institute's summer school, and now we are still developing the socio-ecological multiplex. And I am very excited with the perspective of including an economical layer in this model, because I think it will help us to understand these different dynamics related to health and ecosystem processes. So thank you very much.
Thank you, Cecilia. It's a very interesting presentation. Okay, now the questions.
Brilliant, thanks, Celia. So again, it's only me and Darren that have access to the questions, so we're sort of, you're going to hear a lot of our voices, unfortunately. But yeah, I mean, Cecilia, that was brilliant. Thank you very much. It was just a great example of merging different types of data, using both theoretical and empirical methods, and merging the results of those together. And I think it's exactly that kind of thought process and thinking that we really need to start to merge together and start to really achieve the aim that we're suggesting here in this symposium of merging social, economic and ecological networks. So thank you very much. That was an amazing talk. We've got one question I can see here from the audience saying, well, thank you, I love the work, but they're interested in what type of simulation you used to assess the spread of the parasite.
Which type of simulation?
That's
so
it.
It's a susceptible infected model, and the base, it's a network. It's like a random geographic graph. So it's a geographic graph. You have several nodes in the system, and these nodes, they have information because it informs you what is the species, what are their parameters, and you have this probability of infection if it's in the vectorial layer or in the trophic layer, and this is how the propagation goes. And how species interact. So this is a geographic process because we have, as I said before, the basal geographic layer that determine the species connectivity. So this is why, so depending on the body size, the species have larger home ranges, so they can interact with a larger number of nodes in this environment. If they are smaller, they are more limited in the region, and so in the nodes that they can interact. So this is more or less how is the process of building the networks. So it's basically a geographic process because it's like co-occurrence is what it determines the probability of interactions and if they can interact or not.
That's brilliant. Thank you very much. I do also have a question myself, if you wouldn't mind me asking. You talked about using the large predators or large mammals as potential for sort of monitoring. I was wondering whether you had any thoughts about trying to manage infection, whether there are any points in the network where you could disrupt the flow or the transfer of these parasites.
Yeah.
On that, brilliant.
So this is exactly what we did in those simulations, what we call the immunization simulation. So what we did there was like removing that species out of the system and comparing the dynamics when you have that species or when you don't have it. And then we see that some species, they have a much stronger impact when you remove them. So that's why the infection time, it can be faster or slower when you remove. So then if we have a slower infection time, mean that it goes faster. If it's short, so it's the opposite.
Great, thank you, Cecilia. That's brilliant. Got one more question as we've still got a bit more time, and that's from Anna, and she asks, what interactions between the species in the food web layer did you include?
Okay, I included predator-prey interactions. So these species, they're basically mammals. So it's and this is I got in the literature. What is the diet of those species? So then this we could infer which species they can interact. So which species they can eat based on their diets. And also the vector in this case, because the vector is an insect and there are species that they eat insects. So they were also included in the trophic layer as a resource.
Great, thank you very much. I was wondering, does anyone on the panel have any specific questions? Or not the panel, the group of speakers?
I was just wondering, in terms of the slide that you presented showing the disease linked to the soybean production as well, and whether you could just sort of give us a little bit more detail specifically about that? Is it just the complete collapse of interactions that you might find in a native biodiversity fragment, for example, which causes these sorts of problems?
Yeah. So in the case of the hantavirus, what we know is because it's related to rodents, okay, and it's transmitted by aerosol. So what we know in these places where you have a lot of crop production is that you increase a lot the abundance of some of these rodents, that they are the reservoir of the hantavirus. And what happened also when you concentrate a lot of grain, so you have a lot of resource there, because this is how they keep the soil. So you accumulate a lot of rodents there also. So there's a lot, like high abundance of rodents in this place because they have a lot of resources. And how it's transmitted, they urinate in the place, and then you have aerosol, and then people, when they breathe that, they get infected. So this is how it's related. It's the mechanism. So when we see about like behavioral risk related to hantavirus infection, is very much related to deforestation. So also in the moment that you are removing the forest, this is increasing your contact rate with those aerosols, and then you can become infected. And it's related also to keeping a lot of grains together and increasing the abundance of those rodents.
Thank you very much.